Okay, Anna, we're good to go.
Hey, welcome to today's Open Research webinar hosted by eLife. This series aims to give early career researchers an online platform to continue to share their research as an alternative to in-person meetings, which became impossible now. My name is Anna Akhmanova. I'm a deputy editor of eLife. My own research is focused on the cytoskeleton and particularly on microtubules. But today we will hear talks from three very different young researchers working on Stemness. This will be Ralitsa Madsen, our first speaker, Kevin Eng, who will be talking about evolutionary conserved truncated PDL1 variant, and last but not least, Nikolaos Vakirlis, working on orphan genes. So before I tell you a bit more about how to ask questions, I would just like to very briefly say how important it is that we do these kind of webinars for two reasons. First, to provide a platform for young researchers to share research broadly when meetings became impossible. And second, also because on the news, we read now every day about the importance of science. We're in a unique period of time where everybody turns to scientists for answers and for ways to move forward. And so I'm particularly excited to be able to participate in this eLife webinar so that we can share the newest research with you. So, of course, you will listen, but you will also be able to ask questions. After each 10-minute talk, we'll have 5 minutes for questions, and then we will move to the next talk. To ask a question, you can either type it into the Zoom or directly in the Google document when the link is shared also in the chat. We are joined today by Miranda, Anya, and Naomi from eLife, who are working in the background to support you. They will help you to line up the questions. And if it will be possible, we will invite you to ask your question in person, and then the team will unmute you so that you can do that. Otherwise, if it's not possible, I will read the questions and include your name. The open notes document is also a place for you to contribute shared public notes. We welcome you to do so and to list yourself as a contributor in the list above the speakers to today's webinar. Thank you. Okay, then about YouTube. So this webinar is being recorded. It's also live streaming on YouTube. And since it's a live webinar, we ask you please to be respectful, honest, inclusive, accommodating, appreciative, and open to learning from everyone else. So please behave yourself, don't attack, demean, disrupt, harass, or threaten others. Encourage this kind of behavior. If you feel uncomfortable or unwelcome on any of these webinars, please contact eLife by email. The inbox for this is watched by Miranda at eLife. And organizers reserve the right to ask to leave and to deny access for subsequent webinars on Zoom. If you need any help at any point, please send Miranda, Naomi, or Anya a chat message directly using Zoom. Okay, so now that we are through the introduction, we will start with the first talk, who is from Ralitsa Madsen, and she will tell us about the regulation of stemness. Welcome, Ralitsa.
Thank you. I'll just try and share my screen now. Thank you. So can you all see this? Right. Thank you everyone for joining in, and particularly eLife for what I think is a really great opportunity given the current situation. So I've never given an online talk before, and in case something goes completely wrong, I want to make sure I thank the right people at the start, particularly my previous supervisor, Robert Semple, well, can't go back, but I want to thank my previous supervisor, Robert Semple, with whom I did my PhD at the University of Cambridge and subsequently a postdoc at the University of Edinburgh. Several other people have been involved in this project, also my current mentor, Bart van Hezenberg at UCL Cancer Institute. Now, I'm going to focus on the big picture here, looking at stemness and how it's regulated by PIK3CA activity threshold. But for those of you who want more detail and an actual look at the data, you can come back to this slide on finding the slides online, and you will be able to find all the data upon which this talk is built. So going forward, the focus of my research really is the PI3 kinase signaling pathway. And for those of you who are not familiar with this pathway, all you need to know for the next 10 minutes is that when it's activated in a cell, that cell is told to grow and to proliferate and to resist apoptosis. So quite unsurprisingly, this pathway is commonly hyperactivated in cancers. And often this is because of an activating mutation in the PIK3CA PI3 kinase alpha enzyme. This enzyme is a lipid kinase composed of a catalytic and a regulatory subunit. The catalytic subunit is known as P110 alpha, and that subunit is encoded by the PIK3CA gene. Cancer biologists will be familiar with the PIK3CA gene. It is the second most commonly mutated gene in human carcinomas, and the mutations in this gene are activating, they're considered cancer drivers, and therefore a lot of focus has been placed on the development of PI3 kinase alpha inhibitors for use in oncology. Now, having said this, it is also very important to stress that PIK3CA mutations, or activating PIK3CA mutations, should not automatically be equated with cancer. So we know that this is not the case from a group of rare overgrowth disorders, commonly referred to as the PIK3CA related overgrowth spectrum, or PROS. So this lady here has extreme lower body overgrowth caused by an activating heterozygous PIK3CA mutation that has been acquired during development. And in this particular case, this mutation, although it also occurs in cancer, it does not lead to malignant overgrowth. So you've got a situation here where you can have the exact same activating PIK3CA mutations, but in different contexts and leading to different outcomes. So when I joined the Semple group to do my PhD, my aim really was to study activating PIK3CA mutations in a developmental context, because we knew a lot about them already in cancer, but we knew relatively little about their consequences in development. So to do this, I chose to use pluripotent stem cells. And of course, as it typically happens in research, you end up studying the least, what you didn't plan to do. So ultimately, I ended up learning a lot about PI3 kinase alpha in cancer and contributed perhaps very little to PROS understanding. The reason for this is that pluripotent stem cells share many similarities with cancer cells. And in fact, you can view cancer as featuring aberrant activation of embryonic programs. And in fact, this is now supported by big data sets where, for instance, the research study derived a molecular stemness score from pluripotent stem cells and used that stemness score to then classify human cancers as either differentiated or undifferentiated. And the researchers were able to show that the higher the pluripotent stemness score in these cancers, the more aggressive and the worse the disease outcome. And this duality, if you like, came in very handy in my research because I were able to study the effects of oncogenic PI3 kinase alpha activation on stemness. And I've put a question mark here because this is typically not a phenotype that is widely studied when we think about PI3 kinase signaling. And that is perhaps something that we should start paying more attention to, as I'll come back to later. So as I said, initially, I wanted to develop human pluripotent stem cell models of PROS. So I use CRISPR-Cas9, and the idea was to knock in the strongly activating PIK3CA H1047R variant into human-induced pluripotent stem cells. So in hindsight, I turned out to be very lucky because my CRISPR was quite efficient and I ended up getting homozygous as well as heterozygous mutants. And I say lucky because this is an oncogenic mutation, and the general view at the time was that you only really need a single copy for its full-blown effects to be observed. So in other words, an extra copy was likely to be redundant and, if anything, not particularly relevant for what we wanted to understand. We now know that this was a wrong assumption, even when just looking at pathway activation, and again, this is all published data, we were able to discern graded activation of the PI3 kinase signaling pathway going from wild type cells to heterozygous H1047R mutants and then to the homozygous counterparts. But the most striking difference between homozygous PIK3CA in mutants and the heterozygous and wild type cells was really the effect that this extra copy of PIK3CA H1047R had on stemness. We found that homozygosity, but not heterozygosity for PIK3CA H1047R, resulted in a phenotypic switch characterized by self-sustained stemness. So in other words, these cells are now resistant to differentiation. And we think this is quite important also in the context of cancer, as I'll return to later on, because we also demonstrated that you can have multiple PIK3CA mutant copies in human tumors, and therefore this dose-dependent regulation of stemness and perhaps other phenotypes downstream of these mutations could potentially also be relevant to look into in cancer. On the mechanistic side of things, we of course were very interested in trying to understand what maintains this switch in the on position. So what is maintaining the self-sustained stemness? And importantly, is this a reversible switch? Can we bring it back off again? And is it simply a matter of using one of the PI3 kinase alpha inhibitors that is being developed for use in oncology? So we took an unbiased approach here in collaboration with Rune Linding's lab in Copenhagen, where we profiled our cell lines using RNA sequencing and also proteomics, and consistently our computational predictions suggested that the activation of the TGF-beta signaling pathway plays a key role in this self-sustained stemness phenotype of phenotypic switch that we observed in homozygous mutants. This caught our attention for several reasons. So very early on in my work, I had observed that homozygous H1047R mutants exhibited a strong upregulation of this gene here known as NODO. So developmental biologists will know about NODO as being a critical morphogen during embryogenesis. It's a TGF-beta-like ligand, and like TGF-beta, it instructs cells to undergo epithelial to mesenchymal transition. And during embryogenesis, this is very important for gastrulation and organ development. And again, exemplifying this duality between cancer and development, TGF-beta in a cancer promotes metastasis and disease progression. An important feature of NODO is that it can actually promote its own expression. So you've got this positive autoregulation, which again is very important considering the self-sustained phenotype that we observed in these cells. The reason why it's important is because you also need to supplement NODO or TGF-beta into the maintenance medium of human pluripotent stem cells to keep them in the undifferentiated state. And we knew that homozygous mutants were capable of maintaining the undifferentiated state even when you switch them to a medium that wasn't promoting this. So we hypothesized that self-sustained TGF-beta nodal signaling could be the mechanism for this stemness phenotype in homozygous iPSCs. So we went on to test this, and I apologize in advance for what may look like a very complicated slide. So I'll walk you through it. It's actually fairly simple. On the left-hand side, I've got results for wild-type induced pluripotent stem cells, looking at NODO expression and also NANOC. NANOC is another very important pluripotency factor. It's a downstream target of the TGF-beta signaling pathway. And if you look at the two bars here, the left one is the gene expression in cells cultured in complete medium. And then when you remove NODO, you now see a decreased expression of NODO itself as expected, and it also translates into lower NANOC expression. The same is not true in homozygous mutants, they remain high in nodal expression. And even when you add a p110 alpha inhibitor, so here BYL 719, although you see a drop in nodal expression, this is not sufficient to trigger downstream downregulation of NANOG and other factors that I'm not showing here. What we needed to suppress this Stemness signature was actually inhibition of the TGF-beta signaling pathway consistent with our hypothesis. So just to summarize, what I've told you about is that homozygous H1047R mutants exhibit this self-sustained stemness, and it seems to be triggered by constitutive TGF-beta signaling. We can inhibit this phenotype with a TGF-beta inhibitor, but very importantly, we cannot reverse the stemness phenotype using a PI3 kinase alpha inhibitor. So for cancer biologists, how is this, does this have any relevance? We think it does, and here is some unpublished data which we're quite excited about. We looked at breast cancer transcriptomics, and we computed a PI3 kinase activity score and a stemness score based on gene expression data, and we saw a remarkably strong positive relationship between the stemness score and the PI3 kinase activity score. And more importantly, this was also related to tumor progression, with grade 3 tumors, the most undifferentiated tumors, having the highest stemness score and the highest PI3 kinase activity score. So I'll finish here by just highlighting two of the questions that arise from this. One very important question is whether PI3 kinase inhibitors will act effective against cancer stemness. This is not something that we typically evaluate when we use these inhibitors. We tend to look at proliferation and growth, but we think that it is also important to look at stemness because you need a single cancer stem cell to survive to actually regenerate the entire tumor. So even if the inhibitor suppresses growth, it might not be effective in the long run. And finally, PIK3CA H1047R and other PIK3CA mutations are heterozygous in PROS, and maybe this is one of the reasons why the growth in these patients doesn't progress to malignancy. I think I might have run over time, so I think I'll stop here, and I'll be happy to take questions. Do I need to stop sharing?
Naomi, do we have any questions from the audience?
The questions are just coming in on the document. Anna, I've got a first question in the chat for you, if you like.
Okay, so a question, pretty obvious one. Could the lack of a wild type allele rather than the presence of an extra mutated allele of PIK3CA be the driver of the observed phenotype?
Yeah, so that's a question that has come up sometimes when I present this work, and we haven't actually looked at this directly, but we don't think it is the case. Partly because there is a negative regulator of the PI3 kinase pathway, P10. When you lose that homozygously, you also trigger self-sustained stemness. So downstream of PI3 kinase, if AKT is hyperactivated in human pluripotent stem cells, you again see self-sustained stemness. So we really think this relates to a PI3 kinase activity threshold, which you can achieve by other means than just the PIK3CA H1047R mutation.
Okay. The next question is from Michael, who may be watching you on YouTube. Have you checked the canonical TGF-beta SMAD, such as SMAD2/3 levels in the PIK3CA mutation?
Yes, and we've done a reverse phase protein array, and we've shown that SMAD2/3 phosphorylation is increased in the homozygous mutants, and that you cannot reverse that with a PI3 kinase alpha inhibitor, even though you reverse the canonical PI3 kinase signaling pathway. So yeah, the canonical SMAD2/3 pathway is on.
I see. Okay, maybe a question from me. So how well do you differentiate stemness from proliferation and these different effects? So how can you define these different phenotypes?
Yeah, so we actually don't have any evidence for these cells proliferate more than the wild types and the heterozygous mutants. So we don't think it is related to proliferation in this case. What we know is that these the mutants, but that's seen both in heterozygous and homozygous mutants, they have increased resistance to apoptosis when you remove the growth factors.
I see. Another question is, which PI3 kinase and effectors do you think would be involved in the pathway that you are looking at?
So in a preprint study that we've published recently, we've looked at this with a network computational analysis, and we think that MYC may be playing an important role. So MYC is being stabilized downstream of PI3 kinase and AKT activation, and we see quite substantial evidence for it playing a role in this, and also partly based on prior literature. But it's something that's a work in progress.
I see. And maybe the last question, do we have more questions from the audience? Not at the moment. We have one more question. So this Stemness switch, how do you envisage it mechanistically?
So, this boils down to a bit of complex signaling dynamics, but what we envisage is that you've got this a bistable state, if you like. So at the transcriptional level, this could, for instance, be caused by epigenetic modification and opening of chromatin, and then you enter a state where you have more open chromatin, for instance, at the stemness genes, and continuous binding of activating factors whilst you're inhibiting the repressors of the phenotype. So we believe that there is, when I talk about the switch, it's basically at the signaling level. If you imagine a steep sigmoidal curve, above a certain threshold, you then stabilize into this new state, and this is how we imagine that this is happening. But as I say, this is something that needs further mechanistic studies. It's not being addressed, or we haven't addressed it at the moment.
Thank you, Ralitsa. This is a very exciting study. So thanks a lot. We now move to our second speaker, who is Kevin Ng, and he will talk about PD-L1 variant functions. Welcome, Kevin.
Great, thanks so much. Let me just start sharing my screen. Great, is that working? Great, okay. So thank you very much for having me today. I hope everyone is staying well in these rather unprecedented times. And I think this is a really fantastic initiative to continue to share science in these times. Our lab is an immunology lab, and we are primarily interested in T cells, which are in many ways central in orchestrating immune responses to cancer, infection, but also can be responsible for autoimmunity and autoinflammation. So a T cell response in its most generalized form begins upon recognition of a T cell's cognate antigen by the T cell receptor, the TCR. And when this happens, this results in clonal proliferation of that particular T cell and release of various soluble mediators, cytokines, and other signaling factors. And of course, this can result in all sorts of collateral damage resulting from an overly exuberant immune response. And so in order to restrict this response and to prevent this collateral damage, T cells express PD-1 upon activation. And what happens upon binding with its primary ligand PD-L1 is that you get a decrease in the magnitude of the T cell activation response. And so the importance of this pathway is highlighted by the phenotype of PD-L1 deficient mice, which develop spontaneous autoimmunity. Conversely, PD-L1 overexpression is a very well-studied mechanism of tumor immune evasion, and there's been remarkable success in the clinic by targeting this pathway. And so these two dichotomous phenotypes to me really highlight the importance of tightly regulating the expression and activity of PD-L1. So what I'm showing here is the gene structure of CD274, which encodes PDL1. So you have the IGC and IGV domains on exons 3 and 4, and a transmembrane domain on exon 5. And we did a few years ago, our lab did established a pipeline to do de novo transcriptome assembly of RNA-seq data from both healthy and cancer patients. And we assembled this truncated variant. And we saw that this truncation occurs due to exonization of a retroelement, a line element within intron 4, which donates its poly-A tail to the transcript. And hence we refer to this transcript as CD274L2A. And biologically, why this is quite interesting is, as I've mentioned, the transmembrane domain is located on exon 5. And when we see that we model the protein that would be translated from either full-length PDL1 on the left or truncated PDL1 on the right, that the truncated transcript retains the IGV and IGC domains required to interact with PD1, but lacks a transmembrane domain. And so we hypothesized that expression of this transcript would produce a soluble form of PDL1, which has previously been described to be detected in the serum of cancer patients and in other pathologies as well. What was interesting to us as well is that this intronic region was evolutionarily conserved among all hominid species, which is shown here in this red box. And when we compared the sequence divergence between all mammals, we observed that introns within the gene, which are in gray, and the non-coding exons, which are in white, show far greater divergence than the coding exons, which are shown in dark blue, and this is of course expected. But what was quite remarkable to us is that this line element, which is shown here in orange, shows comparably low divergence to the coding exons. And so in terms of expression, what I'm showing here is expression of full-length PDL1, which is split by tissue type in either cancer in orange or non-malignant tissue in gray. And generally, we see higher expression in cancer, but we also see expression in non-malignant tissue. And this is primarily in immune tissues and barrier sites, such as the lung. And then when we compare expression of the truncated transcript, this is on average 10-fold lower. I've expanded the y-axis just for clarity. And as with the full-length transcript, we see highest expression in immune and barrier sites, such as the lung and the skin. We didn't quite see the same extent of upregulation in cancer. And surprisingly, we saw quite widespread expression among non-malignant tissue. And so the conservation in hominids, as well as the expression in non-malignant tissue, to us suggested a potential conserved biological function. And so in order to test our hypothesis that this truncated transcript would produce soluble PDL1, we cloned it into an expression vector and transfected 293 T cells, which are deficient in PDL1. And when we ran an ELISA on the supernatant of these cells, we saw a dose-dependent increase in soluble PDL1. Importantly, when we analyzed these cells by flow cytometry, we see no increase whatsoever in surface transmembrane PDL1. And this demonstrates that this truncated transcript produces only the soluble form of PDL1. So of course, the obvious question, what does it do? Of course, our first assumption was that soluble PDL1, like its transmembrane partner, would be immunosuppressive. And so we can assay the immunosuppressive capacity of PDL1 by taking primary CD8 T cells from humans and co-culturing them with 293 T cells transfected with full-length PDL1. We then activate these T cells and then analyze the proliferative and cytotoxic capacity of these cells. So as we can see, what we've done here is we've stained the cells with a proliferation dye, and the more cell divisions the cells go through, the more the dye dilutes. And this is what we see in these distinct peaks. And as we can see, stimulation induces robust proliferation, which is decreased in the presence of transmembrane PDL1. However, and to our surprise, no amount of truncated PDL1 transcript was able to induce any suppression. And this was true even at equivalent DNA concentrations as the ones used for the transmembrane form. And given that we found that the truncated transcript was expressed 10-fold lower in our RNA-seq data, we reasoned that these were already supraphysiological conditions. We've also tested for granzyme, other activation markers, CD4 T cells, and K cells, all of which were sensitive to transmembrane PDL1, but not soluble PDL1. The one phenotype we did observe was a decrease in PD1 staining on the T cells with soluble PDL1, and this could be reversed with the addition of a blocking antibody to PDL1. However, we didn't see any decrease by qPCR of the mRNA for PD-1, ruling out the possibility that soluble PD-L1 was somehow decreasing the gene expression. So what could be happening instead is that the soluble PD-L1 was binding PD-1 and thus blocking the fluorophore from binding, resulting in this decrease in PD-1 positive cells that we see. And this was quite interesting, as it suggested that soluble PD-L1 was able to bind the PD-1 with insufficient affinity to cause suppression, and thus biologically could be acting as a receptor antagonist. And so to test this directly, we did a co-transfection experiment in which we keep the concentration of the transmembrane PD-L1 constant and titrate down the soluble PD-L1, again in 293 T cells. And we generated PD-1 overexpressing Jurkat cells. This is a T cell line, and it's a more tractable system to study PD-L1 mediated suppression. And again, we stimulate and stain for activation markers. So as before, co-culture of these T cells in the presence of transmembrane PD-L1 decreased activation. As we could see, and what we noticed was a dose-dependent, rescue of activation as we titrate up the soluble PD-L1. And once again, when we tested this in primary T cells, we saw that the presence of soluble PD-L1 with transmembrane PD-L1 was able to rescue proliferation and cytotoxicity, and that this phenocopied the effect of adding a blocking antibody. And so, of course, we wanted to test this in vivo, which is slightly complicated by the fact that the analogous transcript and protein does not exist in the mouse genome. But given that human PD-L1 has been reported to bind mouse PD-1, we decided to test it anyway. But we also designed a construct for the orthologous mouse gene in which we truncate mouse PD-L1 at the same position. And so we stably transduced these two constructs into MCA38. This is a colon adenocarcinoma cell, and we subcutaneously injected these into immunocompetent mice. And so what we know is that these cell lines in this subcutaneous model are sensitive to checkpoint blockade, as seen when we inject the therapeutic blocking antibody to PD-L1. But what we saw as well is that cells transduced with either human or mouse soluble PD-L1 phenocopy the kinetics of exogenous PD-L1 blockade, suggesting that soluble PD-L1 has the same receptor antagonist effect in vivo. And so what very quickly, this has all been published in eLife last year, and so this is quickly some of the work that we've done since then. We were interested in the cell type, in the biological relevance, and what we found through analysis of RNA-seq data, that this transcript was specifically expressed in activated CD4 T cells. And we could again confirm this doing a time course PCR analysis of primary CD4 T cells. And in terms of relevance to disease, we are collaborating with Marina Bado's group at Imperial, which has RNA sequencing data from CD4 T cells from healthy donors and lupus patients. And what we see is that full-length PD-L1 is upregulated in lupus compared to healthy donor CD4, with no real difference between active and inactive disease. And in contrast, the truncated transcript is selectively upregulated in patients with active disease. And if we express it as a ratio between the full-length and truncated transcript, we can differentiate quite nicely between active and inactive disease, suggesting that this may be a useful biomarker in this lupus setting. So just to quickly conclude, we detect this truncated PD-L1 transcript in both healthy and cancerous tissue. We know that this truncation occurs due to exonization of an evolutionarily conserved retroelement. The expression of this transcript produces a soluble form of PD-L1 that's not suppressive by itself, but antagonizes the bioactivity of full-length transmembrane PD-L1. And finally, we observe that the truncated but not full-length PD-L1 is expressed in CD4 T cells upon TCR engagement, and that this phenomenon can be observed in patients with active lupus. And with that, I'd like to thank my lab, all of the platforms at the Crick Institute, and so on. And I'm happy to take any questions.
Okay, thank you very much, Kevin. So we're ready to take questions. Do we have any from the audience?
We've got some questions in your chat, Anna.
I see. The first question is, so what do you think regulates this switch between the two PDL1 isoforms?
Right, so that's been a very active, we've been pursuing that question quite actively, and we don't really know yet. What we suspect is that the 3' UTRs of these two isoforms are very different. And what we know from the literature is that the 3' UTR of the full-length protein, it contains many regulatory elements. And so we suspect that it might be some sort of RNA binding protein, and we're actively doing experiments with that right now.
Are you planning to pursue that? Look at which RNA binding proteins bind there, or microRNAs or something like that?
Yeah, so I'm currently doing some experiments just to see, to screen a panel of RNA binding proteins that we know are expressed in CD4 T cells with similar kinetics.
We have a question from Jonas Dutra. Thank you for the very interesting talk. When did this soluble PDL1 appear during the evolution of the hominid lineage?
Right, that's a really interesting question, because as you've seen, this intron is very well conserved among the hominids, but not other species. But what puzzled us a bit is that this class of line elements, these L2A elements, are extremely ancient and would have integrated likely millions of years before the divergence of the hominid lineage. And so what was interesting to us is we were able to reassemble, for instance, the mouse genome and the rat genome, and we were able to see traces of this remnants of this line element that had inserted there, but have since mutated away. So what we suspect is that this insertion itself is very, very ancient, but it has only been conserved in the hominid. And we see evidence in some other species, such as the pig, for instance, which has, where we can't detect this line element, but we see other integrations from other retro elements that seem to sort of do the same thing.
Okay, and then the last question. So what can we do with this molecule in terms of cancer therapy?
Yeah, so again, that's been something we've been pursuing, given our rather preliminary in vivo data showing that it manages to delay the growth of tumors. We think that it might be promising, given that this is a molecule that's much smaller than the blocking antibody and might have better penetrance within very dense tumor tissues. We're also exploring whether we can use this system expressed in CAR T cells, for instance, where you can have very localized and temporally regulated checkpoint blockade. And this might go some way in ameliorating some of the side effects of systemic checkpoint inhibition.
I see. Very interesting. Thank you very much, Kevin. And now we'll move to the last speaker, Nikos, who will talk about orphan genes. Welcome, Nikos.
Hi, thank you very much. Let me go ahead and share my screen here. Is this okay? Perfect. So I'd like to thank the organizers for putting this together. This is a really nice idea to keep science communication alive during these weird times. So the title of my talk is Synteny-based analyses indicate that sequence divergence is not the main source of orphan genes. And this is work that I did while postdoc at the Department of Genetics of Trinity College Dublin. So let's dive right in. So what is an orphan gene? So an orphan gene is quite simply a gene that we cannot assign to any known gene family. So in this toy example that I have here, the blue, black, and red genes are shared between all these species. So these gene families have members in all the different genomes that I'm showing here. However, this green gene that we see in the focal species here is an orphan because we only find it in this species. So we cannot assign it to any known gene family in this simple example. Therefore, it is an orphan gene. And in fact, if we include the phylogenetic relationships between the species that are represented by these toy genomes here, we can easily infer that that the most likely scenario is that this orphan gene has originated, so it has appeared along the branch leading to that species over there, and it was missing from the ancestor of all these species. Therefore, it is an evolutionarily novel species-specific gene. So the concept of the orphan gene can be generalized to include taxonomically restricted genes, which are genes that are not only found in one single species, but that are found in a small group of species, such as the yellow gene that I'm showing here, that is found in the group species B. And these taxonomically restricted genes, we also call them TRGs, and orphan genes are found in any clade that we look at across the tree of life, so they are ubiquitous. And what's interesting to think about is how this richness of patterns of taxonomically restricted genes and species-specific genes evolved from an early ancestor that likely had a small genome with a limited gene repertoire. So this leads us to ask the question, how do novel genes originate? So in general, during evolution. So there are two main ways. The first is through sequence divergence. So what does that mean? Quite simply, if we imagine an ancestral gene, okay, that following a speciation event is going to produce two homologous genes. And with the passage of time, these two homologous genes can find themselves within the twilight zone of sequence similarity. Right here, where sequence similarity between these two homologous genes is no longer detectable. So with the passage of time, we can find two homologous genes in species A and B here that are actually invisible. So we can call these invisible homologs because no detectable similarity is present between them. So another way that new genes, that orphan genes and novel genes in general can result is de novo out of non-genic DNA. So this is when an entirely new gene is going to emerge from scratch from a part of the genome that was previously non-genic, such as an intergenic region. And for a long time, this was thought to be as so improbable that it was virtually impossible. But we now know that this isn't the case because we found de novo genes or de novo emerged genes in any species that we look at. So to sum up, we have two drastically different mechanisms by which novel genes originate. We have sequence divergence, we have ancestral gene that diverges and that produces two genes or gene families where no detectable similarity can be found between them. And then we have de novo emergence, where we have an ancestral non-genic sequence that evolves into a functional gene. I'm going to focus on the first one here, sequence divergence. And the question that we wanted to ask is, can we study sequence divergence beyond recognition directly? Can we study this mechanism of new gene origination directly, which is something that has not been done before? And the second question is, so that amounts to basically identifying cases like what I showed you before. So homologous genes that have entered the twilight zone of sequence similarity, where no similarity can be detected between them. And the second question that we wanted to ask was, can we estimate the contribution of this mechanism, so sequence divergence beyond detection, to the total pool of genes without similarity? So the total pool of novel genes, which includes orphan genes, taxonomically restricted genes. And why this is important is because for a long time, as I told you, de novo emergence was thought to be extremely rare. So the assumed picture was that sequence divergence is going to account for the majority of novel genes, and de novo emergence is going to be very rare. So wanted to ask, is there a way that we can actually test this and find out whether this is true? So what we used to do this is we took advantage of the concept of the property of synteny. So synteny is basically the gene order along a chromosome, and conserved synteny means that there is conserved gene order between two genomes in two different species that we're comparing. So in the example that I'm showing here, we have a block of 4 genes that are in conserved synteny, which we have identified using sequence similarity as we normally do. And then in the middle, we have the yellow and the green gene here, okay, which we don't know if there's any sequence similarity between them. And we're going to go ahead and assume that in most cases, the yellow and the green gene are going to be homologous, because they're found within this microsyntenic block of conserved gene order between two genomes. So they found opposite each other within this block. Now we're gonna, based on this, we can then calculate the proportion of the number of times in where we have this arrangement, but no detectable sequence similarity can be found between these two genes, so the yellow and the green gene. And then we need to take into account that this proportion that we have calculated based on genes that are within this arrangement is going to be an upper bound, because so this is an estimate, and of course there's going to be times where the genes that are found opposite each other in this scenario are not going to be actually homologous, so we need to take this into account. And finally, we can use this proportion, and which we have calculated within genes that are found in conserved microsynteny, and extrapolate it to all genes in the genome. And we can do this if we know that genes that are found within conserved synthetic blocks and outside conserved synthetic blocks evolve at more or less the same rate. So this is the idea, to use the concept of conserved synteny. So we built a pipeline using this concept, and we applied it to 3 separate data sets using 3 focal species, fly, yeast, budding yeast, and human. And we compared it to a number of species that are found in a progressively increasing evolutionary distance from each focal species. So we have a number of pairwise comparisons using these 3 focal species, comparing them to each target species separately. And what we can do with this is that we can see how this proportion that, again, as I'm showing here on the left, is the proportion of genes that have entered the twilight zone of sequence similarity, how this proportion scales with time. So we see that there's a very good linear relationship with time since divergence between the 2 species that we're comparing. So each point here represents a pairwise comparison between the focal species and 1 target species, and we can see that there's a good linear relationship. So this tells us that divergence beyond recognition occurs at a steady rate. But we can see that actually in human, we seem to have a much slower rate than the other 2 data sets. So this is the one takeaway that we get from this. And the second one is, as I told you, is wanted to estimate the contribution of this mechanism to the total pool of orphan genes. So what percentage of the total pool of orphan genes does this account for? And we can do this in the following way. So first, again, we have one proportion that we have calculated within genes that are found in conserved synteny. So in this toy example here, we have this proportion would be 1 out of 6, because in 1 out of 6 cases, there is no detectable sequence similarity between these 2 genes. And then we have another proportion, which is calculated over all genes in the genome. And this is basically the total number of orphan genes, if we take a simple pairwise scenario into account. And now we can extrapolate the first rate that I showed you, so the rate in the bottom, to the entire genome. So this is an estimate that we calculate over a subset of genes in the genome, but we can calculate it over the entire genome. If genes inside and outside synthetic blocks evolve at more or less the same rate, and this is what we found. So I'm going to go ahead and speed up a bit here. So we can extrapolate this rate, and what matters in the end is to compare these 2 rates, and we can do this. We have the 2 rates that we're comparing here. So in the solid bars, we have the proportion of genes that have diverged beyond recognition, and in the transparent bars, the proportion without similarity in general between a pair of genomes. Okay, and if we take the ratio of these proportions, we can see that the estimated proportion that is explained by sequence similarity is actually much, much lower than we would have expected. So on average, this is about 1/3. So 1/3 of the total orphan genes in these pairwise comparisons that we have, of the genes that lack similarity, can be explained by a divergence beyond recognition. So this is how much this mechanism explains. So on average, about 1/3. And I'm gonna skip this last part because I think that I'm running out of time. Sorry.
Yeah, so if you just can wrap up, please.
Yes, so I'm gonna skip this last part. So just to sum up really quickly, so we have a novel approach to detect pairs of invisible homologs based on conserved synteny between genomes. We found that divergence beyond recognition occurs at a steady rate within a given phylum. And then the main takeaway is that surprisingly, we find that divergence accounts for approximately a third of genes without similarity on average across the different comparisons that we have. And then the last one didn't have time to talk about. But if you're interested in more about this work, it was published very recently in eLife, and this is the paper right here in the bottom. And I'd like to end by thanking Aoife McLysa, who was the group leader in where I did this postdoc, and Anne Carbunis at the University of Pittsburgh, so our collaborator in this work. And thank you very much.
Thank you very much for a very exciting insight in the evolution of our genome. The first question is, so how sure you are about the evolutionary origins of these genes? I mean, for each particular type of origin, what is the error margin that you have? And can it also be a different type of origin, let's say horizontal gene transfer or something like that?
Yeah, that's a good question. So we don't really know what the false positive rate, let's say, for one particular case would be. We know that we for sure overestimate the number of times that the genes found in this arrangement, where we have two homologous genes opposite each other but no sequence similarity. We know that a lot of times these might not be homologous, so the actual proportion are likely to be lower than the ones that I'm showing here. So this average proportion of a third might actually be lower. We don't really know how much lower, but in terms of individual pairwise comparisons of genomes, it depends on how far away the genomes are that we're comparing. If there's a big evolutionary distance between the species, then the number of genes in conserved synteny that we can find is lower. So then this percentage has a higher error margin, if that makes sense. So the more the number of genes in conserved synteny that we have between two genomes, the higher the confidence that we can have in this proportion is.
I see. We have a question from Jonas Dutra again. Thank you for the nice talk. He has a question about giant DNA viruses, which are well known to have lots of orphan genes. Could this approach be applied to study these viral or orphan genes?
That's a great question, actually. Yeah, I've read about these huge viruses, and they have huge, like crazy percentages of orphan genes. It depends on whether there's synteny conserved between viruses, and I would suspect that there might not be that much, but I don't really know because I have not really well versed in viral genomics. But it is a very, very intriguing question of where do these orphan viral genes come from, and because viruses evolve so fast, so they mutate so fast, it's a valid hypothesis that they might be the result of divergence. But it'd be really interesting to try to apply that to giant viral genomes. Yeah.
I see. We have one last question. So you have these genes which are homologous, like distant cousins, and they don't have any similarity, but do they still bear some properties in common?
Yeah, so I didn't have time to talk about that, but I also looked at secondary structure properties and composition properties between these between invisible homologs. And we found that in a very small subset of cases, there is correlation over these properties between these different pairs. And this is the case when genes, so these invisible homologous genes, share a protein domain. So the proteins that they encode for share a protein domain. So there's no pairwise similarity there, but if you identify protein domains on both of them, they share a common one. And when that happens, you find that other other properties such as protein secondary structure tend to resemble. That again, in the absence of sequence similarity at the protein or DNA level between these genes. So yeah, there's a small percentage of cases where this happens. And presumably this might mean that they have common function as well. So the function is retained.
So we have one more question from Neil Pratt. Are there any other studies that attempt to quantify the contribution of divergence in the evolution of novel genes?
Yes. So there's a number of studies that have taken a simulation approach, and they have, there's a difference in the simulation approach in that you need to assume that every time that when you do a simulation and you see that you end up with a number of the percentage of orphan genes that are due to sequence divergence, that doesn't really prove that they are due to sequence divergence, but it proves that they could be, which is also important. Now there's these studies that have taken a simulation approach, who have more or less concluded similar things as our study, and then there's a recent paper, a recent preprint, I should say, that is taken a more analytical approach to model how similarity decays over evolutionary time. And in that work, they find that divergence accounts for slightly more than the majority, so slightly more than 50% in their data set. So they give a range, of course, and our ranges and with theirs, they overlap significantly, but their estimates are slightly higher, which is interesting.
Thank you very much, Nikolaos. I think we have run out of time. So thanks to all our three speakers for very different and very exciting talks. Thank you very much for sharing your science. Before we finish, we will run a poll. So would like to hear from all the participants about how we could improve these online talks for you as audience members. We are now launching a poll. Our question is, if we changed anything, which two ideas below do you think we should prioritize? Please pick up 2 items you would like us to prioritize and select nothing. You can also select nothing. We welcome other ideas in the Zoom chat as well. And you can also email us. So we will give you a few seconds to do this.
Anna, one thing to say at this point as well is if there are any attendees who'd like to stay on to chat to the speakers today, we can keep Zoom open. So if you just say in the chat or put your hand up or something, we can just get an indication of how many people would like to do that. I think all speakers are available to stay on for a little bit longer. So we're happy to help you do that.
Okay, so I hope that you have filled in the poll, which would be really useful for us to improve these online experiences. And thanks again, Ralitsa, Kevin, and Nikos for sharing your work. Our online research talk series will continue on Thursday at 5 p.m. British time. Will be chaired by another deputy editor of eLife, Detlef Weigel. So we will hear from these speakers, which are now shown on screen. And so please join us on Thursday if you have time. So for updates about this series, please follow eLife community, and there you can find the schedule and the registration information. And finally, I would like to thank all of you, the speakers, but also all the participants. And once again, you are very welcome to stay online to have an informal chat with the speakers. Thank you very much.
Thank you very much, Anna, and all the speakers. That was fantastic. I'm going to stop the live stream now, but we'll keep Zoom going just a little bit to see if anyone wants to stay.